All right, this is take 2 on trying to go through the test-taking strategies in terms of using a codon chart. First of all, you will notice that I have covered up the question, which is always a good test-taking strategy, so that you can write down all the things that you know based on what is on the paper before you get to the question. And the word codon, that looks like a funky word, but to me, it reminds me of the word code, and a codon actually is the 3 nitrogen base sequence that is going to code for an amino acid. Now, amino acids, you may recall that amino acids make proteins. And for proteins, for a protein, it is the sequence of the amino acid I'm just going to do AA, that determines the type of protein it is. All right, and proteins, you may recall, proteins are found in the cell membrane. Proteins are found in your hair, your nails, muscles, etc. So protein is a very important molecule. It is one of the important organic molecules, but that's another topic. Now, how we make these proteins is that we have a series or a sequence of amino acids. In the codon chart, remember I said that on the codon is a code, and the code is made up of 3 nitrogen bases. So if you look here, first letter of that code, you would choose whichever letter it is based on the code from the DNA, the second letter, and then the third letter. Now, we're going to have plenty of practice with this as we're trying to figure out the answer to the question. The question is, which of these changes to the DNA triplet? This is our triplet. Remember, we had said earlier a codon deals with the 3 nitrogen base sequence. Triplet means 3. All right, and we see 3 nitrogen bases here will affect the protein produced. So DNA has the code. This we know. The code is here. DNA has the code, and that code is made up, again, of the nitrogen bases. Okay. Notice that I'm writing all over around the codon chart. I'm writing on my paper. This is a great test-taking strategy. I call it taking a dump on the test. And what that means is you're going to dump all the information that you can remember about a specific topic that may be found either in the stem for the question or the actual question itself. Okay? So right now, I'm taking a dump on the test. So DNA we have is the genetic code, and it's made up of nitrogen bases. It says that there's a triplet. This code is in triplet form, and we know that here are 3 nitrogen bases, again. And those nitrogen bases are going to affect the protein produced. And we know that proteins, again, are made of amino acids. Actually, long chains of amino acids. And it is the amino acid sequence, again, that is going to determine the protein. Gives you your protein. And again, in your codon chart, you can see there are bases, 1, 2, 3 letters. from your code, and these are amino acids. All right? So, here is my original code, GCT. You might recall that in the cell, in the nucleus is where you have your DNA. But that DNA, that genetic code, needs to make it out of the nucleus to a protein site or protein builder. And what carries that code is something called messenger RNA. And it is going to help to make the protein. All right? So this is our original code. We have to decode this into what it would be for our messenger RNA. So G, the complementary nitrogen base, is cytosine, which is C. Cytosine's complementary base is G, which is guanine. And thymine's complementary base in messenger RNA is adenine. Okay? So now I'm going to come to the codon chart. My first letter is C. The second letter is G, so I'm going to go all the way over to G. And then I'm going to go C, G, A. So the actual amino acid, I have C, I have to come all the way across to G and find A, is arginine. All right? So, now we're going to finally look at our answer choices. Let me slide this up a little bit to make sure that we have plenty of room. So, this is my DNA. It has to be translated into my messenger RNA, which is going to carry that code to the protein site. So what goes with guanine is cytosine. What goes with thymine is adenine. What goes with thymine is adenine. So here is my codon, my code. So I come to C. Next letter is A. C-A-A. C-A-A, did I do it right? Looks good. So it is glutamine. Glutamine is the amino acid for A. For B, notice I'm going to go through all of my answer choices, which is always a great test-taking strategy because sometimes you uncover another answer that is a potential answer. So we don't want to give up on anything too early. We notice that arginine and glutamine are two different amino acids. So far that is potentially the answer we're looking for. I come to T. Thymine is paired up with adenine, cytosine with guanine, thymine with adenine. I go onto my codon chart. I find A, over to G, find A, arginine, arginine. All right. So far this amino acid is the same as this one, and we're looking for something that's going to affect that protein. All right, so here's our DNA. It's got to be carried out to where proteins are made, which is carried by the messenger RNA. Thymine pairs up with adenine, cytosine with guanine, cytosine with guanine. So I find A. My second letter is G. And my third letter is G. The amino acid is arginine. I come to D. This is my DNA. This is my messenger RNA code. Guanine pairs up with cytosine, cytosine with guanine. Now, in RNA, there is no thymine. It actually pairs up with something called uracil. So I'm going to come again, my first letter, I'm going to find C. I'm going to go all the way over to G. Then I'm going to find U, arginine. So now I want to look back up to my question. Which of these changes to the DNA triplet? My original triplet was GCT. This said GTT, that's a change. This says TCT, that's a change. This says TCC, again, that is a change. And GCA. All of these are changes in that DNA triplet. Now the question asks, which of these changes to the DNA triplet will affect the protein produced? If proteins are made up of a sequence of amino acids, that will determine the protein. Then any change to the amino acid is going to change the protein. My original amino acid is arginine. Arginine, is that going to cause a change? No. Arginine, no change. Arginine, no change. So the answer is A, glutamine. Now, an issue with a question like this is that, one, if you find an answer that you think is the answer right away, you stop and not go through the rest of your answer choices. You need to always go through all your answer choices. Two, this question takes a lot of time because using the codon chart just takes a lot of time. You have to remember it's several steps. This obviously is a much more rigorous question, but if you will take your time and be very methodical with your thought process as well as use all the space on your test, you can find the correct answer.